Hi all, welcome to today's seminar, which is organized by the EuroCC2 project. EuroCC2 is a pan-European project with more than 30 countries participating. The goal is to enhance the competencies of Europe in advanced technologies like high-performance computing, machine learning, artificial intelligence, and high-performance data analytics. As part of EuroCC2, we offer a series of seminars and training events, as well as customized services for industry, academia, and the public sector. Today's seminar is entitled Computational Investigation of Underlying Disease Mechanism and in Silico Drug Repurposing, and is given by Professor George Spirou, Bioinformatics Era Chair, Professor and Head of Bioinformatics Department at the Cyprus Institute of Neurology and Genetics. Professor Spirou is a Bioinformatics Era Chair holder and the head of the department at the Cyprus Institute of Neurology and Genetics. He is a bioinformatics course coordinator at the postgraduate school of CING, where he has been elected as full professor. He's a senior IEEE member, and he serves as a member of the editorial board in well-known bioinformatics-related journals. Let me now give a brief summary of today's talk. Network science, together with machine learning and computational modeling, lays out a roadmap for the further development of bioinformatics towards a more efficient exploitation of single level as well as multi-omics. Network-based inference and integration provide great opportunities to develop innovative methodologies that lead to new insights into candidate biomarkers, repurposed drugs, and disease-related mechanisms, supporting and enhancing the vision of network medicine. Network-based methods and tools for mechanism understanding, drug repurposing, and biomarker discovery, developed by the Bioinformatics Department at CING, will be presented. Professor Spirou, you can have this.
Good afternoon. Thank you very much, Constantine, for the kind invitation. I'm very glad to be here with you to present this theme and with this opportunity, our work on this direction. So just to say that at the Cyprus Institute of Neurology and Genetics, we are hosting the Bioinformatics European Research Area Chair funded by European Union, and this group that was generated by European funds then is transformed in a department, a permanent department at CING, trying to serve as a hub of excellence in the fields of applied bioinformatics for early diagnosis and effective prognosis, drug discovery, and through these goals towards preventive, personalized, and precision medicine. Our group, we still work as a group and not as a department, so our name is CBIG, SIG Bioinformatics Group. We try to deal with multi-omics and multi-source data integration, network analytics, highlight complex patterns of potential biomarkers, find candidate repurposed drugs, and generate profiles that we feel that they could be a very good stepping stone for AI systems. We are working with networks and with other techniques. Today, we will see a focus on applications with networks. Networks are quite good to, let's say, represent the complexity of the interrelationships between the levels of the data that we have. So we are working within the layers of data and across the layers in order to generate profiles. And through these profiles, we can proceed by analyzing them to find biomarkers or to find candidate repurposed drugs and to understand what is happening with the molecular mechanisms. Just to say that a network could be considered as a visualization of relationships among a set of entities. So just set the entities and ask a question about their relationship, and you will be able to construct a network. And in medicine biology, we can have many network types, like gene regulation networks, where some genes activate or inhibit other genes. So we can see that there is a directionality in the network. Or we can have protein-protein interaction networks, where some proteins, they physically interact with other proteins, and we have a symmetric network, because if protein A interacts with protein B, then also protein B interacts with protein A. But we may have also some more conceptual networks, like the drug gene target network, where the nodes are together drugs and they are gene targets, or the disease-to-disease network based on the connectivity between diseases, depending on the question of relationship each time. So we may ask about their common genes. We connect diseases based on their common genes or their common phenotypes, their comorbidities, etc. And if we have at hand these networks, then we can proceed with analyzing them by applying all the graph theory methods that we can have, trying to highlight the hubs, or trying to measure all the full set of centralities around the network, local centralities and global centralities. We can highlight modules or communities on the network that are related to the condition that we are investigating. We can try to highlight what would be the effect of a random or a strategic attack on the network, etc. So let's see in a nutshell some of the works that we have done in CBIG. So one of the major problems when we analyze the differentially expressed genes between two conditions, disease versus control, is that by giving these differentially expressed genes, we try and we take a list of pathways that are related to this condition that we are studying. But these pathways are never meaningful in the whole set of the list that we have. And the researchers and the biologists and the clinicians, they usually focus on a small subset of the pathways that are found. What about the rest? So we have developed a method that tries to map all the significant pathways on a pathway-to-pathway network. And this pathway-to-pathway network is constructed by us by mining complete databases like the CING, Wikipathways, Reactome, Gene Ontology, etc. So we are generating a reference network like this one. You see my pointer? No. Anyway, on the left side. And here we see that we have a network where the nodes are pathways and the edges represent relationships between the pathways. Then, by having at hand a list of pathways that is related to our experiment, we are mapping these pathways to the reference network. And we see that some of our pathways, they are connected already and some others are not. So we are bringing the missing links to achieve minimum connectivity. So you see that we can bring extra links that were not significant in our differential expression analysis, but our network topology implies that they are significant to achieve connectivity between the pathways. And this makes us believe that we have a more complete and connected functional story around the significant pathways that we are finding. This methodology has been transformed in a tool, publicly available tool, Pathway Connector. It is published on Bioinformatics Journal, and by Pathway Connector takes as input genes or pathways, and then maps the pathways on the selected reference network, and from there retrieves the missing links and generates connected functional story through graph clustering. Here we have the application of pathway connector to several diseases with focusing on spastic ataxia and on Huntington's disease. And we have also applied this to Alzheimer's disease and to some other diseases, giving interesting results. And passing to a next concept about the application of networks is the concept of whether we can apply and we can combine together a priori knowledge with omics in order to generate a network that could be used further in our analysis. So we have devised a method where differentially expressed genes, specific drugs, specific microRNAs, pathways, specific variants, and perhaps also omics, if available, can be joined together to give nodal content and sub-edge resolution. So there is information that gives score only to the node, so it contributes to the nodal content, and some other, it gives score to the relationship, the pairwise relationship between gene A and gene B. So by integrating all the existing information, either putting the information in a quantitative manner as nodal content or adding this information to have edge resolution, we can generate a synthetic gene-to-gene network. This gene-to-gene network does not exist in the databases. It has been generated by integrating the a priori knowledge and the omics that we have. And what we can do with this. Yes. Yeah. Okay.
Sorry.
Okay, no problem. Mm-hmm. Okay. We're okay? No. Okay. No problem. What can we do with an integrated network that carries the a priori information about the disease that we are studying? Actually, we can use this integrated network as a map to informed walks over another network. So, for example, this method that is called pathwalks tries to identify pathway communities using a disease-related map of integrated information. So you see that by using this integrated network as a map, we are guiding millions of agents through another network that is the path-to-path network. And by summing the over-visited nodes and edges, finally, we can highlight communities that are related to the map. What I'm always saying is that we may have the same physical connectivity, for example, Nicosia, and we have various tourists that come. One tourist may have at hand a map that is related to museums, and then going back to his country, he will describe Nicosia based on the museums that it has. But another tourist may have a map that is related to pubs and clubs, etc. So he will go back and he will describe the same town in a different manner because he had another map. So if we have a map that is related to the disease that we are looking, and if this map is good enough, so it carries all the integrated information that we know up to the moment related to the disease, then we can guide random agents, not so random, actually informed agents, through pathway-to-pathway networks, and then ask for the over-visited regions. And then we will see communities that are related to the model, the map that was driving the workers. These ideas at the time of COVID were most of them applied to a collective effort that we did against COVID, where we collected the information that we had that time. So we had some omics available, some info from the databases, and we integrated them in a gene-to-gene synthetic network. And this gene-to-gene synthetic network was the map that was guiding the agents through the pathway-to-pathway network to highlight communities. Here is the gene-to-gene synthetic network that generated this pathway-to-pathway network in the communities that were highlighted. And we showed that the highlighted communities, they were very well connected to the pathological mechanisms known some months later related to COVID. And this investigation was the background to perform repurposing for drugs against COVID, trying to highlight drugs that they are either targeting the host or the viral proteins. And from the repurposing drugs that we had after our calculation, a group of experts tried to shortlist and bring at a final table drugs that would be good to be tested. Some of them they were drugs that were featuring that time against COVID. So it was an internal validation that our findings were meaningful. In another line of thought, when we deal with progression of diseases, differential expression is not the best way to go. So when we have a disease, for example, colon cancer, and we have stages of this disease, then we can take differential expression genes per stage, and then we can proceed by asking to find the subset of these genes that are also monotonically expressed genes. So we try to find the genes that they have a fold change constantly increasing or constantly decreasing through the progression of the disease. And then we are passing from differential expressed genes to monotonically expressed genes, and from classical pathways to monotonically enriched pathways. And then we have a smaller group of genes and targets to reverse if we would like to stop the progression of the disease. And with this line of thought, we try to find the drugs that they are reversing this monotonicity. We found a group of drugs that they reverse the monotonically expressed genes, and also we correlated them with the monotonically expressed pathways, ending up with a short list of drugs that could be used against the progression of the disease. The monotonically expressed genes and the highlighted drugs, they are related with the literature. So we have a first verification against the literature that they make sense. But of course, we need experimental validation to see whether our findings they are good enough. I always say that bioinformatics they are working behind the scenes. So we are predicting, we are calculating, and then we are collaborating with experimental labs to find whether our findings or parts of our findings are correct. In some cases, we achieve this, and we will see some examples. When we have a group of diseases that they could be and they should be connected in a disease-to-disease network within a cluster, so they are sharing, for example, common genes, or they are sharing common mechanisms or common phenotypes, we expect that there is a common pathological background between them. We tried to investigate this hypothesis against fibrosis. So we tried to collect 13 different fibrotic diseases and collect the publicly available transcriptomic data from them to analyze them separately for each disease and to find the differentially expressed genes and the pathways that are related to the differentially expressed genes for each one of these diseases, and also the repurposed drugs that are related to each disease. And from there, we try to generate subnetworks of disease-to-disease connectivity between these 13 diseases. So for the same diseases, we can have different connectivity depending on the question. So we can connect them based on their similarity, whether they carry common overexpressed or common underexpressed genes. So we are connecting these dots. Each dot is a fibrotic disease, and we're connecting each dot with another on the basis of their commonality of common overexpressed genes or common underexpressed genes, or based on their commonality, based on their common pathways that they are sharing, on their common drugs that they are predicted to act against them. And through this construction, we are finding roots of repurposing. So by focusing on a connectivity property, we can build a hypothesis of transferring drugs from one disease to another. So if we have a drug that works to a fibrotic disease, if this is strongly connected with another, then we can have evidence and we can find the roadmap of connectivity to pass the drug from one disease to another, so to repurpose. If our hypothesis is correct and the networks, they describe somehow the situation of the biological condition that we are studying, then if we have progression of the disease and we have different networks that describe this progression, then by network differentiation and network rewiring methods, we can capture this change. So by measuring the connectivity between the same nodes across the disease progression, then we can highlight relationships as biomarkers and not only entities as biomarkers. So we are going beyond the concept that we have genes, proteins, metabolites as biomarkers, but we try also to capture relationships as biomarkers between them, trying to build complex patterns of biomarkers. And we are working in progression studies regarding Alzheimer's, regarding Parkinson's, and recently we are working also against sepsis with quite impressive results. We try to complete the manuscript that shows that we can have impressive differentiation for ICU patients between survivors and non-survivors in a 5-day course. But again, through these computational calculations, even when we try to give repurposed drugs through the basic concept that there are drugs that they are reversing the profile of the disease, even then we have at the end the same headache. Other lists of drugs they are called repurposed at the time, that we don't know from where to start. So we need to be able to re-rank and shortlist these candidate repurposed drugs. Actually, this is the correct name, not the name of repurposed drugs, because we don't know if we are saying the truth regarding the effect of the repurposed drug, but we are highlighting that they are good candidates as repurposed drugs. So again, we have lists of candidate repurposed drugs, and we need to shortlist and to re-rank. So we try to work in this direction to re-rank the repurposed drugs based on their functional similarity with the. So we try to find whether the targets of these drugs, they are related to the functions that are related to the disease under study. So we try to filter the drugs in re-rank based on their functional similarity to the disease. Also, we try to group them in order to have good representatives. Imagine if we have a list of 50 drugs and the first 20, they belong to the same structural family, is not useful, because another lab will take our list and they will start from the beginning, from the top ranked drugs, and they will try to experimentate with the first 5 or 10, but all they will belong to the same structural family. This is wrong. We need to, let's say, have representatives for all, from all the structural families that we have detected. So we try to group together the compounds and the drugs that we are highlighting and re-rank, so to have representatives from all the families, and so to give chemical diversity and functional similarity in our final list. So we try to give tools that you give the initial list of repurposed drugs, of candidate repurposed drugs to be correct. And then, based on this criteria, there is a re-ranking of them, and we tend to believe this re-ranking and to take a short list out of them. By using tools like this, for example, the one that is called CODRES, and we have developed this, we applied this to idiopathic pulmonary fibrosis, and we found the highly ranked drug, niclosamide, and this niclosamide was further tested from colleagues at University of Rus, Democrition University, and they found very interesting initial results. So niclosamide attenuates inflammatory mediators associated with fibrotic responses in subepithelial lung myofibroblasts. So we have an initial experimental finding that our top ranked, after the ranking drug, one of the top ranked drugs, niclosamide, has an effect against fibrosis under the study of idiopathic pulmonary fibrotic disease. This work never ends, because we try to re-rank better and better and optimize our lists. So in another effort, we tried to give bonus or penalties in the re-ranked drugs based on their structural similarity with drugs that are already in clinical trials in phase 1 or 2 or 3, depending on the level of the phase. We give a different score based on their similarity to existing drugs that they are testing now against the disease, and also with a penalty regarding their similarity to failed drugs. So if our repurposed drug is very similar to a drug that has been failed against the disease, it takes a penalty. And it takes also a bonus if it is similar to the ones that they are now being tested. So we have devised a network-based score scheme, and through this scheme, we tried to analyze and highlight drugs against the 3 stages of Alzheimer's, and from there to generate and to find the pathways that are targeted through these drugs. And you see the colors, they correspond to the different stages of Alzheimer's, and then those are the pathways that are targeted by the drugs. So we ended up with 26 elite candidate repurposed drugs against the 3 different severity stages for Alzheimer's. And we asked ourselves, okay, if we were at 2016 and we could repeat our method with the information that were known then, so the clinical trials up to then, what we could find, and we repeated as an internal verification, we repeated this calculation, and among the drugs that we found, we detected the drug saracatinib as a good candidate, which then entered clinical trials the next year, 2017. So with our method, and by using only the knowledge before 2017 up to 2016, we tried to highlight something that other groups, for other reasons, they highlighted as good candidate to enter clinical trials for Alzheimer 1 year later. Going more and more in the optimization of drug repurposing candidates, now our recent work has a Greek name, almost Greek, Dreamocracy, from democracy, and the R is for repurposing. So we tried and we have generated catalogs through a method that collects all the existing drugs found by or tested by others, either computationally or experimentally in a separate way. We are collecting these lists, and then we are giving resolution to them based on their modes of action, their pathways that they are targeted from the gene targets they have, and their initial indications. So for each list, we have a spanning of in mode of actions, in indications, and in pathways that are related to these drugs. And at the end, we are performing a majority voting. And we see that actually we can have for each disease, Alzheimer's, Parkinson's, Huntington's, and MS, these are the 4 diseases that we worked up to now. We have drugs that they are top score based on the majority vote. So, for example, this drug that is top score for Alzheimer's and also for Parkinson's disease, it is not the case for Huntington's and MS, but we can find that we have top score for Parkinson's here, and for MS here, for Huntington's here, for MS here, for example. So we can generate a reference table based on majority voting that integrates what is the knowledge from experiments done by others. And then we can bring our drug to ask, okay, a new drug that we have at hand, how it can be mapped here. So based on this reference table, can we give a score to our drug? Yes, we can give a score to our drug. And finally, try to give an extra information of the repurposed drugs based on what others they have said and they have found up to now. closing, I would like also to share a recent work that we have done on drug repurposing for Alzheimer's disease through modulation of NRF2 neighborhood. NRF2 is a protein, the corresponding gene that is mainly related to oxidative stress and other related mechanisms. And the community believes that the network around this NRF2 is a very good place for targeting against many diseases, from cancer to neurodegeneration, etc. So since we belong to a cost action, European cost action, that it focuses solely to this gene, NRF2, we worked on this direction to investigate what is happening in the neighborhood of this gene and what is happening in the context of Alzheimer's. So by taking a complex network that has protein-protein interactions and regulation information around NRF2, and by taking also information from Gene Expression Atlas at the European Bioinformatics Institute, taking information regarding the differentially expressed genes found in Alzheimer's cases that are around NRF2 neighborhood, then we generated a list of genes that they are disturbed in the Alzheimer's framework and they are in the NRF2 neighborhood. And we found computationally drugs that they reverse their pathologic transcriptomic profile. And from these drugs, after various optimization and re-ranking methods, some of them, they are similar to the ones that I described before, we highlighted 5 candidate repurposed drugs that they could act against NRF2 activity. And then a group in Spain, they took these compounds and they performed the experiments, and they found finally that the 5 candidates that we highlighted, they activated the reporter for NRF2 activity, both in cell culture and hippocampus-derived cells, having the expected result. So the 5 shortlisted drugs that we name as good candidates against Alzheimer's, they make a story around NRF2 that it is verified in cell lines. So we try to, as I said before, to match our findings and our methods to experimental work, so to have a better evidence of what is happening. Closing, I would like to say that also we try to develop tools. So we try to develop methods. Sometimes these methods, they are going to be good tools, publicly available, like the ones that I saw here. And also, we are applying these methods to specific research questions, either by us or given by other colleagues within CING or from other institutes and universities. I would like to say that currently we are participating to several European grants. I will highlight 2. LMAMI, where we are driving the work package related to data integration. And LMAMI deals with the capture of progression from asymptomatic multiple myeloma to symptomatic multiple myeloma. So we have 3 stages of this disease. It is the MGUS, the SMM, and the MM. So we are from monoclonal gammopathies to smoldering myeloma and then to multiple myeloma. And we need to highlight and to mine omics from transcriptomics, proteomics, peptidomics, epitranscriptomics, clinical data and lifestyle we will integrate them, and we will find biomarkers. We will find candidate repurposed drugs, and also we will combine them with AI methods to have computer-aided diagnostics against this progression of myeloma, hoping that we will find something that either will give a good prognosis, or we will find drugs that will stop or make slower the progression of multiple myeloma. And the second one that I would like to highlight is comfort aids. We are participating also in this European grant that deals with the prediction, monitoring, and personalized recommendations for prevention, relief of dementia and frailty. Our group will work mainly with a local cohort. So at CING, we will collect about 200 patients, not 100 and 100 controls actually. We will have proteomics data and electrophysiology data, hopefully MRI data as well. Then we will integrate and we will analyze them to generate profiles, understand the pathways, the mechanisms, the biomarkers, and hopefully to be able to separate between early onset and late onset patients. With this, I would like to acknowledge our group with our, I believe, excellent researchers that they support all these works and many others that were not shown here. We have several members and associate researchers with us and several PhD students. And if you would like to learn more, we can discuss now, or you can visit our site here. Thank you very much. That's all for today.
It's a fascinating talk. I've got several questions. For the structural information, to what level do you go? Is it 3D or 3D level information, or I guess just the sequence of amino acids, for example, or do you take into account
for the structural information regarding the structural similarity that was shown is based on the SMILES, on the structural format that is mainly 2D, two-dimensional. There are also formats that deal with three-dimensional. The results are shown with two-dimensional. We would like to go to 3D, but cheminformatics is very good, up to our knowledge, and works with 2D fine. We understand the limitation of this of working 2D space, and hopefully when we will reapply chem informatics, we will go to 3D SMILES and structural formats. But we don't apply molecular dynamics and rarely we apply this to our works. We have some works on this, but they are very, very specific. So we have an expertise on molecular dynamics and docking in the lab, but it is not too much.
I see it.
Yeah.
Thank you for the talk. So a more specific question first. So at some point you were talking about, instead of looking at differentially expressed genes, to look at those monotonically. I guess when there is the progression of a disease, you see certain genes either expressed more and more or less and less, right? So how many points in that time progression do you usually have? Like how many steps in the progression do you analyze?
Yes, the results that were shown the time points, they are stages. So we have 4 stages of disease. There is no an actual time point. So we are not analyzing time series data. Now that we are working with another data set regarding Parkinson's, where we have up to 10 years data, we deal with more time points in this manner, and hopefully we will have the opportunity to apply methods that are more suitable to this.
Right.
Yeah.
Yeah, I mean, that was actually at the back of my mind, the reason I asked the question, because if you had a lot of points, we can use methods from time series analysis and do more sophisticated.
We have not, and this is not very easy to obtain omics at various time points. Nowadays, there are data sets, like the Parkinson's data set that I'm discussing, and other data sets. So, and also we have the thought that to apply time series when analyzing neural activity and combined omics with neural activity at several time points. So we are designing to apply time series analytics there, but up to now we haven't.
Then I have a more high-level question. I mean, it's been a few years now that I'm not working in this area, but when I was working on it, it was networks, it was all the hype, all the activity was on networks, and now it seems that the focus has been moving towards machine learning as the main tool for analyzing this kind of networks and this kind of data. I mean, do you have the same opinion that it's moving from network analysis to machine learning as the underlying workhorse of the analysis?
I feel this vibe, and this is actually a trend now that everything seems to be dealt with machine learning and deep learning. I'm not sure whether deep learning will manage alone to solve the problems. I agree that this is a trend. My personal feeling is that a combination between the older fields and the deep learning schemes will be the optimum way to go, because we need to have also explainability in our deep learning schemes, and I think that explainability will be facilitated with networks at least.
So thank you very much. It was very interesting. Just as more for my curiosity. So if I see, well, when you predict all these pathways, for example, you don't have a single pathway in practice. I would expect that this is an inherently, let's say, stochastic problem. So if I see this as stochastic variables, can you predict the probability distribution function of potential pathways?
We could pass to probabilistic modeling with pathways if we try to transform these networks to probabilistic graphs with transforming the scores to probabilities. This could be a very nice work to have some, let's say, predictions on probabilistic graph theory. We haven't done this. It's something that we do more or less through random walks. So the random walks that we have, actually the informed walks, the model-based walks, they deal with these probabilities across the pathways, and finally they assign probabilities to the pathway communities that are related to this. But I think that there is plenty of space in probabilistic graph theory when we transform the static networks to probabilistic graphs.
Yes, just the last point. So if I understand well, you see these features, let's say, of molecular mechanisms, for example, right? And you use them in the network. But is there any way to discover new molecular mechanisms that we don't know currently? I mean, can you use these approaches to discover new knowledge?
Okay, very nice question. We are working with reference networks. So we are working with what is known up to now. So the space of discovering new mechanisms is not so big. But when we are working with link prediction methods, for example, and we see that we can find an unexpected link between two distant mechanisms that we cannot understand how these mechanisms they could be connected, but our algorithm and many others in a robust way, they are predicting highly scored link between them, then perhaps we can suspect that there is something there. We will not be able to name it, but we will be able to expect something, and then we will go to the lab to ask from colleagues to try to see how these mechanisms they are related, and perhaps they will find the new mechanism that is missing. So yes, I think there is space for discovery, but it is limited. Yeah.
Evangelis questions as a follow-up. I was thinking this knowledge acquisition is a dynamic process. So could we perhaps use Bayesian methods for this process where you get new information, so you have to try one?
Sure.
Or is it something that you use? Like, how do you update the networks every time when a new information comes in about new connections, new pathways, new genes? Is it a dynamic process on your existing network, or is it building again?
No, it is a static version. Each time that we are performing the research, we are taking the knowledge up to now. So we have the updated network. It would be nice to have, let's say, various versions of databases that they are through the years. So we will see the evolution of this network construction. And from there, we could calculate probabilities and then predict what would be next. But up to now, the results that we are retrieving, they are based on the up-to-date version of the database that we take the pathways. Thank you very much. Good. Hello. Hello.